Good afternoon, everybody. It's a real pleasure to be here. The last time I was in Israel was when I was beginning my graduate studies with Mark Feldman at Stanford. And I wanted to thank Adi Livnat for inviting me. And it's wonderful to see so many familiar faces. And for that, I can thank the Simons Institute in Berkeley and also Mark Feldman for helping to create this wonderful theoretical community for evolutionary studies. So today I'm talking about interpretability and incomprehensibility in the products of evolution. So organisms have millions to trillions of parts, of whose combined activities emerge replicating structures that behave so as to maintain their organization. That's our best understanding of what living things are. But the basic open questions are: how does organismal structure and function emerge when considered at the full scale of organismal complexity? And how does evolution shape the functional and structural architecture of organisms? These are two principal open questions that I'm interested in. So, in the past few years, artificial neural networks have been created where the behavior is not constructed, but actually also emerges from the dynamics of millions of interacting parts, or thousands up to millions of interacting parts. And now the behaviors rival certain cognitive capabilities of human beings for the first time. And so we now have a second example of complex systems with emergent properties analogous to what living things do. And so the question I'm wondering is, what might be transferable between these domains? So, one of the current challenges of deep learning networks is that their behavior can often be impossible to explain or understand or interpret, and that they can also produce unwanted surprises in their behavior, being fooled, finding loopholes, vulnerability to adversarial examples. And so how can you trust systems you don't understand, can't interpret, and can't explain? So this question of interpretable, understandable, explainable AI, these are all now major research efforts. So the question is, why shouldn't the same phenomena be found in biology? And here I examine the genotype-phenotype map through this lens of interpretability. And so we're going to go through a catalog of processes that enhance or obscure the interpretability of biological processes. And the take-home lesson that I wish to offer is that the degree of interpretability may be the product of the underlying processes and not some inherent feature of organisms or life or biology or chemistry, but should be thought of as a result of processes. So here is a great classical example from Karl Sims, Artificial Evolution for Computer Graphics. So he actually created a system for generating images, and he did artificial selection. He picked the images he liked and then had a system of mutating them and then continued to do selection, and he came up with this great image. Sort of looks like icicles or something like that. And and it was the system that generated it was genetic programming. And in genetic programming, you can represent a function as a parse tree. So here's the Gaussian distribution represented as a tree, and you can take two of them, and you can take parts of them and recombine them, and you come up with a new formula. And that's actually the system that a variation and the underlying genotype that created that image that I just showed. And here is the code that generates this image. All right, it's in Lisp. And if you were to ask how does this create this image, it really is impossible to explain. So, and this is something that Sims noticed early on. He said, users usually stop attempting to understand why each expression generates each image. And so you have this system. This genotype is generating an image. Karl Sims is selecting on the image, and you come up with this. And how this maps to this is completely obscure. And yet, this is what he liked, and this is what he got. So, in nature, You have something, you have in the case of the peacock's tail, which I notice is on one of the posters for the conference, you have the peahen evidently selecting on the pattern generated by the peacock's genome. And the question is, are we going to run into the same problem? So are we going to, is the genome, the genetic coding for this peacock's tail going to be something like this that's completely incomprehensible, or is it going to be interpretable and understandable? And as far as I know, nobody knows the genetics of the peacock's tail as of now. So that's an open empirical question. So in other examples of evolutionary computation, which is now applied to all sorts of things, if you've ever flown in a Boeing 777, the original engine for that was produced by an evolutionary algorithm. Here they tried to get a nozzle that would do a fine spray of paint, and they started out this, and they evolved the shape of it, and they ended up with this as the final shape. And why does this produce such a great spray of particles? Who knows? Here's another example of trying to evolve wires into an effective antenna, and it was the same thing. It was done in a computer and then built, and this was the shape that was the best for the antenna. Why does this work so well? Who can say? So these are examples of products of an evolutionary dynamic that are not interpretable, understandable, or explainable as to why they work so well. And here's an example from a dissertation of a student at San Diego who was trying to use genetic programming to produce code for an autonomous vehicle. And the student actually has the code in the appendix of the dissertation, and there's page after page after page after page of this code, and that works, that drives the autonomous vehicle perfectly well. But the question is, would you get into a car that was driven by this code? So again, as a product of evolution, this is not interpretable. And so for generating artificial intelligent agents that we put trust in, it's a problem, because how do you trust something like this? So a folk psychological definition of these terms, which I'm using fairly interchangeably, I'm sure they have different usages in the literature, but interpretability, understandability, explainability is essentially having some kind of hierarchical decomposability, if you will. And there's actually an interesting paper by Wolfson et al., Have Your Spaghetti and Eat It Too, Evolutionary Algorithmics and Post-Evolutionary Analysis, where they're trying to take some of these evolved genetic programming code and find some kind of way of interpreting what is doing what in the code. And it's of mixed success. So it's also a question that arises in evolutionary computation. Now, the molecular biology program, who's a molecular biologist in the audience? A few of you, okay. So you tell me if this rings true or not, but that the whole paradigm of molecular biology has been that one attempts to hierarchically decompose the phenomenon because that means you understand it, and then understanding is necessary to achieve prediction and control. So there was a seminar at the Santa Fe Institute, and this guy gave this talk of this very complicated model, and Murray Gell-Mann was in the audience, and I feel sorry for the guy, because at the end of his talk, Murray Gell-Mann said, "What is the point of studying a complex system we don't understand by making a complex computer model that we don't understand?" And I thought, oh my God, what a devastating critique. But in retrospect, in this era of deep learning networks, I would say that understanding isn't everything, and that a computer model can be incomprehensible but accurate, and thus allow prediction and control, manipulation and intervention. So what about an AI system? If it seems to work but we don't understand it, should we trust it? So here's an example of some deep learning systems that seems to be interpretable, And in this example of a convolutional deep learning network, you have these different layers that seem to, the activations of the different neurons in the layers seem to be attributable to different properties that we can recognize of the inputs after they're trained. But in other cases, the general question of how do the particular weights in a trained deep learning network generate the correct behavior? Well, people are not even, at least the last time I checked, not even attempting to say why do these particular weights generate this function. Rather, the current explanation is the process that produces the weights, the stochastic gradient descent, is how you explain its behavior. But there are some surprises, these adversarial examples. Now, in yesterday's talk, there was, people showed the example of the two pigs. Who saw the picture of the two pigs? So one of them had this imperceptible noise added to it. It looks just like the same exact pig, and yet the deep learning network says instead of it's a pig that it's an airplane. So this is the exact kind of complementary adversarial example where so this paper, Nguyen et al., what they wanted to do was they wanted to take these off-the-shelf deep learning networks that have been trained to recognize different images and see if you evolved a pattern that would be classified by the network as a certain, like a penguin, would it look like a penguin? And so they actually took an evolutionary approach and evolved pixels to be highly classified by the pre-trained networks as various different animals. And what they found was, so this with 99.6% certainty, the network thought this was a robin, thought this was a cheetah, thought that was an armadillo, a lesser panda, a centipede, et cetera. And so, and to our eyes, it looks completely like static. So this is an example of a false positive where the network thinks for sure that that is a jackfruit, but to our eyes, it's obviously not. So the whole vulnerability to adversarial examples is an area that's still trying to be understood in deep learning networks. So we can't really fully explain how they're working. Now, so what I would like to propose is the following, that biological phenomena range over a spectrum of interpretability, and that different levels of interpretability are primarily the result of different evolutionary processes. And that biology has advanced primarily by working its way from the bottom of the ladder of interpretability upward, and that modern techniques may be propelling us into biological phenomena that may be intrinsically incomprehensible, in that they can't be hierarchically decomposed, and that we need to be ready for that possibility. So here, let's go up this ladder. So from some of the earliest discoveries, the lac operon, this wonderful feedback system of regulating the production of lactase that was easily, I would say, fairly readily understood and explained. You can explain and understand and interpret what all of these parts are doing. Now, we get it to a little bit more difficult and a more recent phenomenon of RNA folding and the structure of RNA. And we get into a little bit even more complicated with trying to understand development. So here is an example of some circuitry, developmental circuitry of a dynamic system that produces these the segment determination in Drosophila. And these days we get into these connectomes, interactomes, and this is an example of the protein interactome this was from 2006, and here it is 7 years later, and we're getting these what are called hairballs, and the question is how do you interpret or understand or explain how these things are producing the organism. And what I'm proposing is that something like this may be inherently understandable, but when we get to a certain level, we may be encountering phenomena that can't be hierarchically decomposed and that really can't be understood any better than why do the particular values of weights on a neural network allow it to play Go better than any human. So I want to go through a small catalog of processes that would seem to confound interpretability in the product of evolution, and then go through a catalog of processes that promote interpretability. So the catalog includes the things that confound interpretability: infinitesimal pleiotropic gene effects, and defensive complexity, and constructive neutral evolution. And then on the other side, processes that promote interpretability: dimension reduction from dissipative dynamics, and mechanisms that select for modularity. And then in particular, the area that I contributed to is how gene origin dynamics contribute to modularity and possible interpretability of the genotype-phenotype map. So let's first go through processes that contribute towards incomprehensibility. So the basic, what's called the infinitesimal model of quantitative genetics, is that genetic variation at many loci have very small magnitude of effects over many traits. And this has been empirically developed recently by Boyle, Pritchard, and company, and they're what they call the omnigenic model. And so the idea is that many parts participate in often unmeasurable degrees in generating many traits, and such a system will not be hierarchically decomposable. That is how it works, will not be interpretable. So as a little kind of visual metaphor, this is a photograph of a remote road on the island of Maui, where I used to live. And when you go out on this road, it looks like there's thousands and thousands of potholes that have been repaired over decades. That's how it looks. So the idea as a metaphor for this infinitesimal model is that a gene comes along and patches a little particular pothole here, but it generates a slightly smaller pothole somewhere else. And then another gene will patch that pothole, but it'll leave a smaller pothole somewhere else. And so what you have is that tens, hundreds of genes collectively patch each other's potholes and make a smooth navigable road. But that trying to understand how a particular gene is contributing, causing this to be a drivable road is no longer, you can't decompose it into anything. So in fact, the main model of quantitative genetics has this property. A second idea, really interesting, that I learned about at the Simons Institute meeting where I first met Adi and Leslie Valiant, is Eric Chastain and company, a model of the immune system, which is to say why is the immune, when you look at the immune system, here's a little example of showing the dynamics of the immune system. There's all these different types of cells, all these different cytokines interacting with each other, and it seems to be unnecessarily complex. And what they propose in this paper is that it's actually defensive complexity, that it's evolving to a point of complexity to thwart pathogens from being able to evade the immune system, to be able to figure out how to get around it. So this is an interesting idea, and in which the complexity is actually a kind of encryption, if you will. And a third idea is what's called constructive neutral evolution, a term introduced by Arlen Stoltzfus, that among all the genetic encodings of a particular phenotype, the more complicated ones may outnumber the simple ones. And so if the different encodings are mutationally connected by neutral networks, which is to say a mutation happens which preserves the phenotype, and so there's no selection against it, then over evolution, the more numerous complicated ones will predominate. And so this is basically an entropic process on neutral networks, and exemplifying what McShay and Brandon have called biology's first law, which is a kind of increase in entropy. So here's a particular example, which is this example of scrambled genes, which is these ciliates have these bizarre genes that are all, so here's the sequence on the genome, but when you look at how this transcript, which is, and then the polypeptide that is finally produced from it is topologically mapped to this, it's all chopped up in pieces. And the hypothesis here is that the ciliate evolved some way so that once there was an inversion on this gene, and it evolved a way of putting it back into the right order, but once it had that mechanism, it kept getting more and more scrambled, and every time it was able to unscramble it. And since there's many more possibilities of having a scrambled ordering than a single linear ordering, it simply evolved into this scrambled gene configuration, where there's no fitness advantage and it's not there for any function whatsoever. It's just that there's more ways of doing it this way than there are of that way. So that's this constructive neutral evolution is another thing that leads to incomprehensibility. And this idea actually was first developed in the field of genetic programming and from a 1997 paper. So what was discovered in this method of genetic programming, where you have a tree that is that's a parse tree of a particular piece of code, and you're doing these subtree exchanges, is that when they actually applied selection to these things, that the average length of the tree would get longer and longer and longer and longer as the number of generations proceeded. And it was a mystery. It's called bloat. And it's a problem because then you spend a lot of time computing things that are actually completely useless for the actual computation that the tree is doing. So one of the proposals is from Langdon and Poley that in general, variable length allows many more long representations of a given solution than short ones of the same solution. Thus, in the absence of a parsimony bias, we expect longer representations to occur more often. And so representation length tends to increase. So that again is an entropic process on the space of equivalent solutions of a problem. And you simply move into the more probable regions of the space where the programs are longer. So that's a little bit of a gallery of processes that contribute towards uninterpretability, incomprehensibility. Now let's look at the opposite direction, processes that contribute to interpretability. So one of them is dimension reduction. So the many-to-one property of genotype-phenotype maps may reflect something mathematically fundamental about the composition of natural functions. So, in fact, dimension reduction is a generic property of infinite-dimensional dissipative dynamical systems. So, you may have what's called the embedding space, may be very high-dimensional, but your sample distribution may actually fall along a much lower dimensional subspace of that. And here's an example from the early days of dimension reduction algorithms. So here, each of these images is really 4,000 dimensional image data, but in this paper by Tenenbaum et al., that's the embedding dimension, but the actual variation was really just along 2 dimensions. So one was closing or opening the fingers. Actually, that's, let's see here. Yes, that's this dimension. The other is rotation of the wrist. Okay, and so there's really only 2 dimensions of variation here embedded in this 4,000 dimensional space, and their algorithm was able to actually properly embed it into a 2-dimensional space. So we find, here's an example of an infinite dimensional dynamical system, a partial differential equation, and even though the dimensions are infinite, when you look at the global attractors of these infinite dimensional dynamical systems, they are typically finite dimensional. And you actually have this nice theorem which says how this lambda parameter, so it has a nice diffusion term, and its change in time is equal to this diffusion plus this function which has certain regularity conditions, and the dimensionality of the attractor is bounded by lambda to a power. Okay, so once you have regularity conditions on this function, you get an infinite dimensional dynamical system gives you a finite dimensional attractor. So, and this is a very generic property. So, so it's a hypothesis that would seem to be reasonable is that organisms, the actual space of their multidimensional phenotypes is really a low-dimensional subspace of of all the different variables you look at when you see the organism, and that this will contribute towards interpretability. Now, the second phenomena is the evolution of evolvability. So the early works on this question, so the generation of adaptive heritable phenotypic variation, okay, is a way of describing evolvability. Because if all you're generating is deleterious variation, the evolutionary process will reach a mutation selection balance and stop. And so adaptive evolution requires some production of phenotypic variation that's in the adaptive direction. So the question is, how can you randomly alter a complex organism and improve its function with non-vanishing magnitude at non-vanishing probability? So a general answer is by diverse mechanisms that focus variation in directions with adaptive opportunity while suppressing variation in harmful directions. So here, a general way of quantifying these ideas is this concept of the distribution of fitness effects of mutation or other kinds of variation operators. So here we have growth rates, fitnesses along this axis, and probabilities along this axis. And if you have the exact same fitness as your parent, then the offspring maps here. So all of these, this region all is deleterious mutation. All the weight at this neutral line, that's mutational robustness, if you will. These are mutations that have no effect on fitness. And then evolvability comes from this upper tail of the fitness distribution. So we can then take this distribution of fitness effects and map it to these classical concepts of genetic load, the more recent concepts of mutational robustness and evolvability. And so that was for a particular, that was for the yeast heat shock protein, 90, and we seem to see this very generic behavior from other proteins, other genetic variation. So here is for an E. coli beta-lactamase, and again you see a mode at neutral evolution, then a large chunk at deleterious, and then this little spike at lethal mutation, so it's typically bimodal, and then this small upper tail of the fitness distribution that reflects evolvability, the ability to actually improve on the phenotype. So if we go back to a classical model of Ronald Fisher, what's called the geometric model, where basically you have a space, a phenotype space, and these spheres are your contours of equal fitness, and evolution then simply random variation moves you towards the center of the sphere, and if you look at the fraction of the variation that's an improvement, that fraction, that upper tail, only goes down as adaptation proceeds. So how can evolvability ever evolve to increase? So clearly it can't in this simple model. So there's a variety of evolutionary mechanisms that have been identified that actually produce an increase in the evolution of the upper tail of the fitness distribution. So typically these models have a recurring variation in natural selection. So it changes from selecting for one phenotype to another phenotype and back and forth. And you can think of that as that alternating selection tends to wear trails, as it were, and that the genotype evolves to places where it can quickly evolve with a very small number of mutations between the selective peaks. So I should mention this Kashdana Ilan at Weizmann, and so one of the pioneers on this. And so here's a depiction of this model. So you have, these are two neutral networks. So these are all the same phenotype A and phenotype B. And if you keep alternating between selecting for phenotype A and then for phenotype B, the genome will evolve to a point where it's poised between these two networks, where a simple mutation can get it to switch from one phenotype to the next. And so that's evolutionary dynamics. So this will tend to then, whatever those genes are that allow the quick change between these two phenotypes, then you can interpret that there are the gene controlling the phenotypes. So that this tends to lend itself towards more interpretability of the genotype-phenotype map. And here is the paper from Kashdana and Ilan. And so they looked, when you had constant selection, you get an uninterpretable genotype-phenotype map. But when you switch between what they call modularly varying goals, then you get to the situation where just changing the connections of a couple of these gates will allow you to switch between the two different Boolean functions you're selecting for. And here it's a general open question, is what mathematical properties of the fitness landscapes allow re-evolvability to evolve under recurring variation in natural selection. Here's another paper, very interesting one, by Jeff Clune and company, where they suppose that there's actually a fitness cost to every connection between the objects, between the items, and this selects for having lower connectivity, more sparse networks, but what they find is that the actually sparser networks have higher evolvability and faster adaptation. So this is a really interesting outcome. Here's another example by Tamar Friedlander, who is at Hebrew University in Rehovot, and she worked with Uri Ulan on and other authors. And so it's imagine a gene or a signal processing network which has inputs and outputs, and where your input space has different ranks of variation. And what they find is that under an evolutionary dynamic, when you have a low rank in your variation of your input space, you end up with a low rank, a very small number of vertices in the middle, so that, and that as you increase the rank, you get the number of internal vertices tends to match the rank of the inputs. And so this allows you, so it's not just a completely connected network that evolves out of these processes. You get a relationship between the variation of the inputs and the structure of the network, which then you could say gives you more interpretability. And here is Adi's contribution, which is the idea of mixability, and that, and I hope I represent this adequately, that sexual reproduction and recombination cause alleles to mix with the other alleles at the same locus and with the genetic variation in the rest of the genome. And this selects for alleles that have high mixability with the standing genetic variation, and modularity in the phenotypic effects of allelic variation contributes to mixability and is therefore selected for. And so this then gives you also a more interpretable genotype-phenotype map with this modularity. And as mentioned in a previous talk, this is where Srivastava And Hinton and company got the idea for the technique of dropout in deep neural network training. So now I want to come to the particular area that I've worked on, which is how gene origin, dynamics of gene origin contributes to a more interpretable genotype-phenotype map. So a basic idea is that genes have two fundamental properties. So we're used to the idea of the generative properties. How does the gene product actually, how is it employed to generate the organism? But there's another aspect, which is the variational property of the gene, which is how does variation in the gene map to variation in the phenotype of the organism. So genes not only provide materials for the organism, they also provide degrees of freedom for varying the organism. And so selection on the incorporation of genes in the genome therefore selects on the dimensions of variation in the genome. So here I want to use an analogy to help understand what the consequence of that is from World War II. So this was the Royal Air Force bombers in World War II were having high attrition at the hands of the German anti-aircraft artillery. And so they did a study of when the airplanes came back from their missions of where the holes were from the artillery. And because they figured they could add some extra armor to try to reduce the attrition of the airplanes. And so they looked at the distribution of the holes on the airplane, and so they were going to recommend that they put armor where the highest density of the holes were. But one of the fellows working on the team was Abraham Wald, and he wrote a paper and he did a statistical analysis, and he said, no, that would be exactly the wrong thing. You need to put the armor where there aren't any holes. He said the reason there's no holes there isn't because they're not being shot there, it's because the planes that get shot there never made it back. All right? So that was his insight. So the planes that got shot here, you never counted them among the returning from the mission because they never returned. And so the analogy then is in the genome, so the sampled planes, they were all very special. They were the ones that survived. And in the same way, the genes functioning in the genome are very special. They are the ones that survived. And so their histories are conditioned on a small choice of common events. That when they were created, they subsequently reached high frequencies in the population due to selective advantage or drift. And the new sequence was preserved because deletion or loss of the function would be deleterious. Okay, so the genes that you look at in the genome have these as conditions on their presence. And so we can ask them, what are the missing holes in the genome? And so some of the missing holes, the things that you don't see among the genes in the genome, are disturbing functions that under stabilizing selection, for example, dosage, and having basically just being neutral additions, that those will be hit by loss of function mutations and eventually disappear. And so the genes that are in the genome are the ones that survived, and which means their immediate effect is advantageous or neutral, and it goes to fixation, and then subsequent mutations give NeoFunctionisation or subfunctionalization or escape from adaptive conflict. These are particular mechanisms that make a new gene basically important for the organism. And so we come to this question of how do new parts get incrementally added to a complex adaptive system. And so I have a computational model that illustrates how selection on gene origins will give you actually very special genotype-phenotype maps. And this is an adaptation of Kaufman's NK landscape model, which we heard earlier. And so the idea here is you have a genome, and this gene will affect some certain subset of the organismal functions. And if this gene mutates, then basically it resamples the functional effect from a uniform distribution independently of what it was. So that's called the House of Cards mutation model. And so now we add a new gene, and it has some randomly chosen effect on this subset of functions, and we ask what happens if the new gene is only kept when it increases fitness, what happens to the distribution of the functions that it affects. And so the fitness of the organism is just the average of these 5 values on the genome X. And so here's our genome growth algorithm. So we add a new gene to the genome. We obtain its functional effects randomly from a distribution. If it produces a fitness decrease, you throw it away. And if it produces a fitness increase, you keep it and then adapt the genome through allelic substitution to a fitness peak. So these are some of the dynamical assumptions, strong selection, weak mutation. And here's what we get from the model. So here's our, when we create our new de novo genes, this is our, reflects the distribution of effects on the different functions that we're drawing from. But when you look at the genes that are actually kept by evolution, you start out just randomly accepting them, but as adaptation gets higher and higher, you get selection for fewer and fewer connections to the functions, for more sparsity. And if you look at the distribution, so this is the, you can call this the pleiotropy. So the number of different functions it's affecting is the pleiotropy. And if you look at the average pleiotropy, it starts out 20, which is the sampling distribution, but as the genome grows, it gets smaller and smaller. And so you're getting a more and more modular genotype-phenotype map. And here's the actual distribution of pleiotropies. So this is the sample that we're, when each new gene is added to the genome, this is what we're, the sampling distribution. But when you look at the genes that are actually kept by evolution, as the gene number gets up, the concentration gets more and more focused on genes that have very sparse effects. And again, so instead of getting this omnigenic genotype-phenotype map where every gene affects every function, you're getting much more concentration of function on a small, every gene later in the genome growth affects fewer and fewer functions. So, but the effects on the adaptive landscape go even to a deeper level. And what we can do is we can take the outcomes of these, take the fitness landscapes that we've evolved and reinitialize them and actually look at the attractor landscape, the attractor distribution. And so here, if we reinitialize the genome and let it evolve on this landscape, and then we look at the fitness of the attractor where it ended up at the fitness peak, we see that these landscapes that evolved through this process of selective genome growth have vastly higher fitness peaks than what we would get from a generic landscape with the same pleiotropy model. And we also see these very large domains of attraction compared to these, which are very small. So the process of conditioning the genes in the genome on the fact that they had to produce a fitness increase when they were added has profound effects on the adaptive landscape. So all of these models need to be refined, obviously, with greater population dynamic realism to make them of interest to population geneticists. But I think the basic phenomena will remain that the selective filtering of genes as they come into being should enrich the genome with genes whose variation is channeled towards adaptive opportunity, and that this may make the genotype-phenotype map more interpretable. So let me conclude. So this is a cursory survey here, but I think it's enough to suggest phenomenology that should be explored. So the interpretability of biological systems or artificially evolved systems may itself be an outcome of different evolutionary dynamics. So evolution on neutral networks, constructive neutral evolution, and the generic properties of dynamical systems, dimension reduction, diverse processes producing modularity, and gene origins filtering for modularity, and other processes that need to be added to this catalog, I think all may shape the extent of interpretability of evolved products of evolution. And to finally conclude, it's an open problem of how to combine these different phenomena that enhance or thwart interpretability in biological systems. And that experience with evolutionary computation tells us to prepare at least for the possibility that the generative properties of the genotype-phenotype map of evolved organisms may, in some cases, be intrinsically incomprehensible. And instead, what we may need to do is understand the processes that produced the incomprehensibility. And finally, to note that interpretability isn't everything. The computational models of incomprehensible phenotype generation may yet be feasible. So thank you for your attention, and I'll take any questions.